Endonucleases make cut at specific positions within the DNA. Now, each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, example GAA-TTC, palindromic sequence, it will bind to the DNA and cut each of the two strands, both the strands of the double helix at specific points in the sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA, example GAA-TTC. EcoRI cuts between G and A on both the strands. You see, on both the strands between G and A in the palindromic sequence. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and this can be joined together end to end using DNA ligase. So even they have the sticky ends, they do need the DNA ligases. Restriction enzymes cut the strand of DNA a little away from the center of the palindrome sites, but between the same two bases on the opposite strands, example between G and A little away from the center of the palindrome sites. There is one thing that unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created, because these would create complications. So it's better to do with the same restriction enzymes. Now, separation and isolation of DNA fragments. There is technique gel electrophoresis. The cutting of DNA by restriction endonucleases results in the fragments of DNA. Now these fragments can be separated by a technique gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them towards the anode under an electric field through a medium matrix. Nowadays the most commonly used matrix is agarose gel, which is a natural polymer extracted from seaweeds. Now DNA fragments are separate according to their size through sieving effect provided by agarose gel. Although the smaller the fragment size, the farther it moves. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. That means you cannot see pure DNA fragments in the visible light and without staining. It needs to be stained with ethidium bromide and followed by exposure to UV radiation. Like later you can see bright orange color bands of DNA in ethidium bromide stained gel exposed to UV light. Now the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. There is spooling, another step. This one is elution, spooling, that is chilled ethanol adding, separating the DNA. The DNA fragments is purified in this way. Now these are the wells, largest one near the wells, then DNA bands, the smallest. So this was the separation and isolation of DNA fragments. There is gene cloning,